All right, so I would like to thank everybody contributing material to this video. So my question to you is, given all of the challenges of HuBMAP, what do you think is your group's greatest contribution to HuBMAP so far?
So we've done a lot in the first year and a quarter of HuBMAP. I think our greatest accomplishments are that we've built what we call the Ingest portal, which is what the tissue mapping centers use to contribute data. And that has now been used. We actually have data now, and that's a tremendous accomplishment. It's been a long time coming. And more data is coming in almost daily now that it has begun.
So with all that data coming in for all of these different research groups and tissue mapping centers, how does HuBMAP address the challenge of integrating such a diversity of data types?
Different groups in the biological front are generating different types of data. And when I say different types of data, they might actually measure the same thing, but using different technologies. So the pipelines that we currently support are basically divided into different modalities and include right now pipelines for the analysis of single cell expression data. So these are the levels of genes within single cell ataxic data. So this is the accessibility of DNA in single cell in different organs or tissues. We have pipelines to support spatial information. So spatial is the location of specific proteins and where they are expressed within a cell and between cells in specific tissues. And the last thing we worked on is image microscopy data and connecting that to something called a mass spec, which can measure levels of metabolites or proteins. So all of these data types are currently supported by pipelines we developed for HuBMAP. And as I said, no matter what platform or technology you use to generate that data, the outcome will look the same for all of these modalities.
So if I wanted to contribute data to HuBMAP, or if I just wanted to explore data from donors and organs that have already been registered, how would I go about getting started with that?
Once you log into the Ingest portal, you see that there are two parts. On the left is the HuBMAP ID system. This is where research groups and other data providers go to first register a donor or tissues extracted from an organ. On the right is the HuBMAP Data Ingest, and this is where data providers upload the data associated with those tissues. Now let's enter the HuBMAP ID system. Here, data providers can create a new entity, either a new donor, that is an individual whose tissue has been donated for research, or a new tissue block sample, that is a tissue sample that can be sliced into sections for preservation and analysis. We'll click on donor. Here is where data providers would enter the information. So for instance, there's a place for protocols.io DOI. This details exactly how the tissue was extracted. And there's a place to enter in any metadata associated with that donor as well. This is where an ID for the donor would be generated. We can also search through donors and samples that were already registered, and our searches can be filtered by research group, donor, and organ type. If a tissue is one extracted from the left or right kidney, it can be registered using the Registration User Interface, or RUI. Then the data is ready for submission. Next, let's look at the data ingest. Here is where researchers can upload data associated with a tissue sample. This creates a landing zone in Globus, a research data management service that provides high-speed, secure file transfer and sharing. Depending on the type of data, there can be specific requirements about what data is able to be uploaded and what naming schemas to use, etc. The data then goes through a review process, from new to processing, sometimes to invalid, but eventually data is published. After data is registered, uploaded, and processed, it becomes available via the HuBMAP data portal shown here. If we select donors, we arrive at a faceted search interface where we can limit our search by research group or creator. Here we can see all the data that this particular creator uploaded. Then you can click on a particular donor. This allows us to search either by the samples or by the data sets that were derived from that donor. Choosing samples updates the search interface to show the tissue samples derived from that donor. And if we look at an individual sample, it will have an interface similar to what we've seen for individual donors. We can now also look at data sets. Let's choose one from the University of Florida. For some data that's been uploaded, there are visualizations. This is one, a HuBMAP plot from a salmon RNA-seq output. The HuBMAP portal lets you download individual files so you can run your own analyses. But you can also use the search interface and the basic visualizations that are available without having to download anything. Now, finally, I would like to show the Ccf User Interface, which has been integrated into the portal. You can access it via the CCF link on the main menu page. This version of the CCF UI uses the HuBMAP Search API, which lets you search for tissue blocks that have a RUI location. You can then explore those tissue blocks in their spatial context together with additional data, including sex, age, and BMI, all uploaded via the Ingest portal.
So now, moving beyond the nuts and bolts of how it operates, what would you consider one of the most unique features of the HuBMAP data portal overall?
I think one of the unique features of the HuBMAP data portal is the tight integration of the curation process with the data analytics, the pipelines that are used to process uploaded data sets that come from the tissue mapping centers in the portal, all the way to the visualizations that can be displayed directly in the web browser, which makes it really easy for an end user to start interacting with HuBMAP data. There's no need to install tools to download any data. You open your browser, you type in the URL, or you pick a data set, and you can start interacting with it immediately. I want to make sure that people will come and say, HuBMAP is a gold standard for how projects at this scale of this complexity should be set up. We want to make tools that people want to use.
I think HuBMAP is unique in that it's bringing together medical experts and data science experts in a way that I have not seen before. We're looking at all tissues in the body at a cellular and subcellular resolution and bringing together what are really state-of-the-art approaches in handling data, handling pipelines, handling computation, and making it very easy to use single portal to get at all of this.